Okay, so today we are starting a new chapter, chromosomes, structure and their function. Okay, so I've given the heading and the road in inheritance because that is basically their function. Now, if you look at the chromosome, I think you have done meiosis or mitosis. You have seen how they look like, no, in the microscope. So you can observe them when you stain them at the metaphase stage, okay, of mitosis or meiosis. Then you stained with what stain? You used safranin, no? Or what was it? I think someone may have remembered already. The name of the stain is acetocarmine, no? So you stained it with acetocarmine and you could observe those chromosomes at different stages of mitosis or meiosis. Then they were rod-shaped, weren't they? And they were paired, no? They were rod-shaped. Okay, so let us see what is written here. They are rod-shaped, dark-stained bodies seen during the metaphase stage of mitosis. And we stained them with a suitable basic dye and viewed under a light microscope. They were first described by Strasburger, okay, in 1815, and the term chromosome was first used by Waldeyer in 1888. So since the 1815, they were first described, and they got their name in the end of the 18th century, 1888. So later on, as the scientists kept on studying it, they realized that the chromosomes are made up of thin chromatin threads called as chromonemata, which undergo coiling. Okay, so this is what you observe in the microscope. You observe this, but each of these is actually like this, super coiled. Okay, super coiled. Highly supercoiled, okay, highly supercoiled. Thin segments that are so coiled that they become very compact. So if you stretch this out, it will be very long. Okay, it'll be supposed like really long. And this is not even half, not even 1/3. Okay, so it's like, you just try, you have a piece of cloth or a chunni, no? This is your long chunni, you squeeze it, it becomes a short ball. So all of this is carrying information. So much information, but you compact it by coiling and twisting so that it occupies the least amount of space. Okay, just like when you travel, when you go on vacation and you pack your suitcase, no? This is, let's say, your suitcase. If this is your, let's say, your t-shirt, you fold it in such a way that it becomes very small. You roll it up, then it takes only this much space. From such a big shirt, only this much space. Then you can fill many t-shirts, then many pants, many jeans, many dresses, right? So when you coil things, when you coil them, coil, C-O-I-L, when you coil them, they become compact, okay? They take up less space. So this is your travel advice also. If you want to travel light, if you want to travel with just one suitcase, you roll up your t-shirts, you roll up your socks, they will take a lot less of space. Okay, now looking at the chromosome number. So how many chromosomes do humans have? How many pairs do they have? So I think you already know the answer. We have 42 total number of chromosomes and we have 23 pairs. Okay. So the number of chromosomes is constant for a particular species. I have 42 total chromosomes. A dog or a cat will have another number. Then oats will have another number. Wheat will have another number. Bacteria will have another, no, bacteria is a bad example. Pteridophyte will have another number, then grass will have another number, and so on and so forth. So every species has a different and a constant number of chromosomes. So in the gametes, the number of chromosomes become reduced, okay, or reduced into half, or they become haploid. So when you have two sets of chromosomes, one is one set, now this one set, this is diploid. Then if you have three sets of the same chromosome, like this, then we call it as triploid. Then if you have four sets of chromosomes, you will call them as tetraploid. Then you can go on hexaploid, heptaploid, and so on and so forth. Now the haploid set, okay, if you reduce the number of chromosomes, suppose I start with a tetraploid, I reduce it into half, there will be two pairs left. So this is its haploid set, even though it's twice n. Okay, even though it's diploid, the half is the haploid set is just 2n. Okay, so this 2n is known as your genome. Okay, means if an individual is twice n, n is the genome. If an individual is 3n, this cannot be used as an example. If an individual is 4n, then twice n is the genome. Okay. So the haploid set of chromosome is known as the genome. So how do the diploidy comes? Okay, diploidy comes when the genome of a male and a female gamete, they fuse. So it gives rise to the diploid genome, okay? Or they get the diploid set of chromosomes. The number of chromosomes of each somatic cell is the same for all members of a given species. The organism with the lowest chromosome number is the nematode. So this is just sort of a net question, no? Organism with the lowest chromosome number is the nematode. Ascaris megalocephalus univalens, which has only 2 chromosomes in the somatic cell. So something very interesting. Ascaris megalocephalus univalens has only 2 chromosomes in his somatic cell. Then in the radiolarian protozoan, Olacantha is found a diploid number of approximately 1600 chromosomes. Among plant, chromosome varies from 2n is equal to 4 in Haplopappus gracilis, this is a member of your sunflower family, to 2n is equal to or greater than 1200 in some pteridophytes. So isn't it weird how the higher plants have lower number of chromosomes? Like these beautiful flowers that we see, the Compositae family, they have less number of chromosomes, and the lower plants, the pteridophytes, the ferns, they have so many chromosomes. They have around 1200. Okay, so this is a table showing you the number of organisms, and the number of chromosomes. Now the first one, this Arabidopsis thaliana, this one, no? When you go for your masters, you'll see this name of plant everywhere. Why? Because all the studies about the genome, about the chromosomes, about the DNA was done in the plant Arabidopsis thaliana. Okay, it is a diploid plant and it has a total of 10 chromosomes. So it has 5 in chromosomes in its haploid set. So you think about it, if you study human chromosomes we have 42, then haploid set 23. Then imagine how many genes out of 23 chromosomes, if you study only the haploid, imagine how many years, because the genes are there in tens of thousands. So if you study only five chromosome and whatever number of genes here, let's say 100, here 200, 300, 500. Yes, it is a big amount to study how this react with this one react with this one react with this. But it's still just 5, no? As compared to the human genome, which is so big in number. So that's why the first example I gave was this Arabidopsis thaliana, which has only 10 chromosomes, okay? And this is widely studied, okay? Till now, all the genome experiments are done using this plant because it has less number of chromosomes and they are constant, and less number of genes and less number of combinations, no? You have done right in school, combination, permutation, probability, no? That this is the gene which causes this. So out of the thousands and tens of thousands of genes, if you have only 5 chromosomes to concentrate, then at least you can map out. Okay, so there are some more examples. Wheat is a hexaploid, has 42. So you divide by 6, means the set is how many only? 7, right? I don't know. Then tobacco is a tetraploid, 48. Then you divide how many it will have in the haploid phase. Then oats is hexaploid again. So these are some more examples. Fruit fly has only 8 chromosomes, so it also has been in the zoology department. Therefore, they concentrate on a fruit fly when they study. We botanists, we concentrate on Arabidopsis. They concentrate on the fruit fly, no? That Drosophila, no? That one is common fruit fly because it has less number of chromosomes.